this video, we're going to learn about how nerve impulses work. In order to do so, we must first understand the parts of a neuron. First, we have the dendrites. Dendrites are short branch extensions of a nerve cell along which impulses received from other cells as synapses are transmitted to the cell body. Next is the cell body. This is the nucleus containing central part of a neuron. Up next is the axon. An axon is the long thread-like part of a nerve cell along which impulses are conducted from the cell body to others. The axon is wrapped in what is called a myelin sheath. Myelin sheaths insulate neurons so they can send electrical signals faster and more efficiently. Also wrapped around the nerve fiber is Schwann cells. This is what forms the myelin sheath of peripheral axons. Note of Ranvier are gaps in the myelin sheath of a nerve. Finally, we have the axon terminal. This is the button-like endings of axons through which axons make synaptic contact with other nerve cells or with effector cells. In order to demonstrate a neural impulse, we've used various arts and crafts items. Here's our movie legend. As you can see, we have acetylcholine esterase, acetylcholine-gated sodium channels, calcium ions, potassium ions, sodium ions, voltage-gated calcium channels, voltage-gated potassium channels, and voltage-gated sodium channels. How does a neural impulse work? Explanation of the process. A large number of voltage-gated ion channels are found in the cell membrane of neurons, which allow for the movement of potassium ions, sodium ions, and calcium ions into and out from the cell during an action potential. When a stimulus depolarizes the membrane to the minimum threshold of activation, voltage-gated sodium channels are the first ion channels to open. As sodium channels open, some sodium ions rapidly enter into the cell. Voltage-gated potassium channels open in response to the depolarization caused by sodium influx. The movement of potassium ions from the cell repolarizes the membrane back to its negative resting potential. In the neuron, voltage-gated calcium channels are only found in the presynaptic terminals. Activation of voltage-gated calcium channels allows the movement of calcium ions into the cell. Calcium entry triggers the release of stored acetylcholine from vesicles in the presynaptic cell. A synapse consists of the presynaptic terminal of one cell opposed to the postsynaptic membrane of the next cell. A small gap between these terminals is called the synaptic cleft. Acetylcholine is released from the presynaptic terminal and then binds to and activates acetylcholine-gated sodium channels, which allows for sodium entry into the postsynaptic membrane. Acetylcholine is rapidly degraded and inactivated by acetylcholine esterase in the synapse. As you will see, the whole process takes place all over again. And that's it.